is a junior cycle experiment in that it's going to be exceptionally unlikely that they are going to ask you to do the whole of this experiment, because as I say, it's past junior cycle experiment. They're not going to want to do it. So if we had been in school, I would have been showing you the anaerobic jar. So this is the one where the control has all three conditions. It has the water, the oxygen and the warmth, and that is the only one that's going to germinate. So you will have a scenario where you have no water, and that's relatively straightforward. So when we do this now, we do them in the Petri dishes. So we have cotton wool in here, and she, I think, said she used radish seeds. We use cress. It doesn't matter as long as it's a really fast germinating seed. They don't actually mind what you use. You can tell them whatever you want, but usually we do it in a fast germinating seed. So this will have oxygen, and it'll have the warmth. There's an optimum temperature for plants to grow. Can you remember what that temperature is when we did the plant chapters?
25.
Yeah, 25 would be probably your optimum. You wouldn't go any higher because you're going to start doing denaturing. So good job, Andrew. So we do no water, no warmth is straightforward in that I can't spell warmth. We put this into the fridge. So again, you'd have your Petri dish set up. This would be your cotton wool. You'd have your seeds, and this one would be in a fridge. So at 4 degrees, we would have the oxygen present, and we would have water present. So that would get those ticks. And it's the last one is the one that they will usually ask. So it's a mixed, it would be in one of the little mixed questions, and it would be a part to it. And they either ask it from the context of where would you use an anaerobic jar, or where would you use a thing called alkaline pyrogallol. So if you can think of like my soda stream bottle as being the anaerobic jar, it's roughly about that height, and it's a fraction a bit wider. And you know the lids that you put on and you clip them on the top, so you get them in like IKEA, and you put your pasta, your breakfast cereal in, and you know they have the little glass lid, and you lock it down. This is the exact same type of thing, and it's a completely sealed container, and what you do is you stack your Petri dishes inside in this container. And so the container is called the anaerobic jar. And if we had done it in lesson, I would have put a chemical into the anaerobic jar. So it comes in a little container like this, and it's called alkaline pyrogallol. And this removes oxygen. It's like a scrubber. So they like to ask, what is the scenario in which you would use alkaline pyrogallol? So this removes oxygen, and the anaerobic jar would be the same scenario. I didn't write that very well, so I'll write it over here better. Alkaline pyrogallol. So I have it highlighted down here in yellow in terms of the question. That would be the thing that they would most likely ask. So you would use an anaerobic jar and the alkaline pyrogallol. So as I say, they won't, chances are of them asking you to do the full experiment exceptionally unlikely. It will usually be part of a question, and it will be, if it does that, it will be looking for, it'll either have these words in the question, or it will be looking for these words in the answer. If they were to ask you the full experiment, you can do the junior cycle, test tube, cool boiled water, layer of oil. They will accept that. So from the video, they asked, why did you use a number of seeds per plate? So Zara, you're the next head on the list. Why do you use a number of seeds?
In case some of them don't germinate.
Yeah, so so in case some don't germinate, you then will still have a result. don't germinate. So the control plate has all three conditions. It gets the full VAMI, so water, oxygen and warmth. And obviously water is moved from its test plate, it's just not added. And how else is this plate treated? It has oxygen and it has 25 degrees. So for the oxygen plate, it's removed, if you said that, and the plate will be a tick for water and it will be a tick for 25 degrees Celsius. And the temperature is reduced by placing in a fridge, so there's at roughly 4 degrees, and it is a tick for water and a tick for oxygen. Okay, happy to scroll to the next page? Yeah, unless somebody shouts. So then in terms of germination, this will get a tick and everything else does not germinate. So for the second experiment, they will specifically always give you both types of agar that you could use, and you can answer using whichever one you want. When we go back to school, it will be the starch agar is the one we're going to use. I will still give you the result for the skim milk plate. So we use some of the skills here, even though you haven't done the fungi experiment yet, but we use some of the same skills in this experiment that we do in the fungi experiment. And we now also have enzymes coming into it and digestion coming into it. So enzyme that breaks down starch. Jess, you're the next head.
I'm not sure, I'm not going to get that one.
Okay, so that is amylase. Remember, we have it in our mouth. And Jazz, can you remember what amylase breaks starch down to?
Maltose, then glucose.
Yeah. So, good woman, it is maltose and then glucose eventually. So, in her experiment, what seeds did she use? Where am I in the bed? James, you're up next.
I don't know, was it? I'm not sure.
They're pretty big ones. So, we're going to use broad bean seeds. They're really, really big. They're about, for the ladies, where your knuckle is on your thumb to the top of your thumb, they're about that size. So why did we have to soak the seeds, Alicia? Did you get that?
Was that to denature or?
No, so this is at the beginning. It's not the boiling. It's you literally just shove your seeds into water. Has to do with what we did the last day in terms of...
God, can't remember, sorry.
It's alright. So we put the seeds in water because we want to start germination. So this is breaking dormancy. We want to start germinating. We want to activate the enzymes because this whole thing is for, we want digestion to actually happen. So we need to start the embryo growing again and activate the enzymes. Robin, we boiled 2 of the soaked seeds. Why do we do that?
To denature them.
Yeah, so we want to denature the enzymes in there, and these are going to be the control seeds, because they are not going to do digestive activity, so they will be the control seeds for this experiment. And now we have get to the idea of why are we now disinfecting everything. So Clara, any ideas? So we'd be squirting the bench, we'd wipe the outside of the seeds.
So no bacteria or fungi grow in the plate.
Absolutely, okay. So we're making sure everything is clean, and that the only thing that's going to digest our starch is going to be the bacteria, the enzymes in the seed. So this up here is what's called an aseptic technique. So it is going to try and prevent contamination. So our agar and things will be sterile. They should have no microorganisms, and we use aseptic techniques to prevent contamination by, I'm just going to write shorthand, microorganism. And the whole point is that it will be the enzymes only doing digestion. I'm going to make this slightly bigger so I can write smaller. Enzymes only digesting. Nothing else on your plate is going to be responsible for that. So then we have to split our seeds in half. How do we go about splitting our seeds? Felon.
Using a sterile blade.
Good man. Okay, so we are going to use a sterile blade. So again, we're not introducing any microorganisms if that's going to be possible. Okay, and why then do we open our agar plates, Millie, ever so slightly, and we place again the seed halves inside with the sterilized forceps?
Prevent microorganisms or bacteria from getting in.
Absolutely. So again, this is another aseptic technique, because they might ask you, give some aseptic techniques that you used. And again, this is for prevention of prevents contamination with microorganisms. I can't spell. With microorganisms. So why do we place the seeds going downward? So I meant to steal something from downstairs. So I'm going to steal my daughter's little stamper. So this is the fluffy side on the stamp, or this is the side with the little ladies in it. Okay, and this is the seed coat at the back. Why is it that I am putting my seeds with the ladies facing the agar, the inside of the seed facing the agar? Concave, any ideas? So that's the inside of my seed. This is my seed coat. Why do I put the ladies onto the plate?
So it can germinate.
So it can germinate. What are we trying to do in this experiment other than germinating? What is the thing we're looking at? Go back to the title and have a look.
That's for starch.
So yeah, so we're looking for the enzymes are all on the inside of the seed. There's going to be no enzymes on the seed coat. So by us placing it onto the agar, those enzymes are in direct contact with the starch, so they can access the food that's there. So the reason we do that is so that the enzymes in the seed can access the starch in the agar. And your plates are incubated. They will be incubated lid facing up, and you leave them in there for about 2 to 3 days, and it will be at the 25 degrees. So in terms of our plates, we will have 2 plates. I'm going to draw them sideways, and I'm going to draw them looking head on. So in terms of our sideways, this is our starch agar, and we would have 2 seeds, So this will be the testa, and the seed is going to be facing down. And this will be our unboiled seeds. And we have the exact same setup, except with boiled seeds. So that's it if we were looking at it sideways. And again, this is our starch agar. And if we could see, when we look down, we would see like this. So the labels all stay the same. This is going to be your testa, but these are unboiled, so this is your test, and these are boiled, your control. So in this one over here, your enzymes are going to be digesting the starch. And we're going to have to do a test to prove that the starch has been digested. So I'll move this up slowly. So where are we in our heads? So Laura, did you get to how far, or what happens when the plates are tested for enzyme activity? How do you test them?
I didn't realise you had to take things down when we were looking at the videos.
Dope. Mark?
I didn't get that far.
Hannah? How do we test for starch when we do food tests?
Iodine.
Was it the colour, not like the result?
Yeah, so but how do we get the result? So if to test for starch, we have to look for we use iodine. So we're going to add iodine. So so this is usually the rule is, if you do microbiology, you never open your plate ever, because you don't know what's growing on it, and it could be terrible, and now you've put it into the atmosphere. In this one we do. So what we do is we remove our seeds from both plates. We pour in iodine, or we cover the plate in iodine. We wait, and then we remove the iodine. So we only draw for our results the looking down of the plate. So if we have starch present, it should go the blue-black colour. So which of the two plates, if this is my test and this is my control, and what I'm going to do is I'm going to just draw in the outline of a seed. So what should I see in my control?
Starch present.
Starch present. So if I literally cover my plate in iodine, and I'm going to do it in a blue. If I cover my plate in iodine for the control, there's been no digestion because there was no enzymes, so the whole plate will literally go blue-black because starch is going to still be present. And if I get any clear areas, then I must have some type of microbial contamination. And in reality, if I was working in industry or in a research lab, I would have to repeat my whole experiment again. Okay, so the whole plate here will go blue-black because starch is present. Starch present, and that's because there were no enzymes to digest. So what do you think I would see if I poured the iodine onto my test plate?
4 white patches.
You should see white patches. So while I'm going to draw them here that it's only the outline of the cells, actually more the digestion, the enzymes kind of move into the agar and they'll go around. But you will see clear patches, 2 clear patches, definitely where the seeds were. But you may also see them kind of around the cells because the enzymes will spread. And the clear patches tell me what?
Starch is not present.
Starch is not present. Okay, so I will get I get clear patches and there's no starch as was digested. And the blue-black is the exact same as the other side. There was no digestion, so blue-black as starch still present. So in terms of the results for each plate, the control is fully blue-black. As no digestion. And the test has clear areas where starch digestion occurred. Does a curd have 1 hour or 2 hours? I think it has 2 hours. Now, if you prefer skimmed milk, you can absolutely use that. The SEC don't care as long as you have it. So the only difference, if you use skim milk, you're testing for protein. So can anyone remember the food test for protein? The name of the reagent, the colours?
Bioresh.
Yeah, so it's Bioresh reagent is used. And what are the colours? So the control plate will be all protein. So I remember what colour Bioresh goes if there's protein present.
Purple.
Yep, so it'll go purple or lilac in colour. And in our test plate, instead of clear areas now, what are the colour of Bioresh if it's not present, if there's no protein?
Blue.
Yeah, so the test, there'll be no protein where digestion happened. And it will be, so there'll be blue patches in purple. So if you want to give yourself a little diagram. So we will this time go blue patches. Ooh, pretty colours. Purple. So as I say, when we finally get back to school, we will do the the starch agar plates. So this is showing protein digestion. And this is our test plate.